AQA GCSE Core Science Unit Biology 1 Set A Higher Tier. Based on the results of a study, a nutritionist claims that a high protein, low carbohydrate diet helps people to lose weight. 200 people took part in the 6-month-long study. Half followed a high protein, low carbohydrate diet and half continued their normal diet. The participants were weighed before and after the study and were also asked general questions on their lifestyle. These are the results. On average, the people who continued their normal diet lost 1% of their body weight. On average, those that followed the study diet lost 4% of their body weight. Name one health problem that's most likely to occur in an obese person, rather than a person with a healthy body mass. So we've got a number of options here: diabetes type 2, heart disease, high blood pressure, arthritis, and you must say some cancers, not all cancers. And that's just for 1 mark, you just say one of those. So can you be sure that the claim made by the nutritionalist is valid? Explain your answer. Well, our first point is to say that more people on the study diet took more regular exercise. You were given that information up at the top there. This could have caused a weight loss rather than the diet. So what you're saying there is that in answer to the question about validity, which is, as you know, a How Science Work term, you need to look at this idea of controls. Was it a fully controlled study? No. Why? Because there were more than one thing affecting the variable. And you know that that's not proper scientific study. Okay, question number 2. Antibiotics can be used to kill bacteria and to help a patient fight disease. Scientists developing antibiotics were testing the effectiveness of 3 different antibiotics, X, Y and Z, against the bacterium. They grew the bacterium on agar jelly in 20 petri dishes, forming a bacterial lawn on the top of the agar. So this looks something similar to an investigation we did in class. They took 4 identical discs of paper and soaked each of them in different liquids, containing, as you can see, the antibiotics. Okay, and number 4 was distilled water, obviously your control. The discs were then placed onto the agar in each dish, and the dishes were kept in an incubator at 35 degrees C for 2 days, and then they were examined. So here's a typical dish. So what you can see here, and they annotate it for you, the black bit represents the bacterial lawn. Now I know lawn seems like a bit of an odd term to use, but that's basically a layer of bacteria that's growing over the dish. Okay, then you've got these bits around the outside of the discs that are completely clear. So you know that whatever's on the disc is affecting the rate of growth around the disc, and it's obviously clear on this one which one is more effective. So let's have a look at the question. What was, which antibiotic, X, Y or Z, was the most effective in killing the bacteria, and explain your answer. Now it hasn't asked you for which dish, it's asked you for which antibiotic. So let's have a look. Obviously here you've got a really big area cleared, which means that the antibiotic is having quite a big effect. So we're going up to the table, we're looking to see it's antibiotic X, and that's our answer. Why? Because it had the largest cleared zone. B. What was the dependent variable? This is a term that you should be familiar with. We've done a lot of controlled assessments now. The dependent variable is what you measure, okay? And what were they measuring to see the effectiveness of the discs soaked in the antibiotic? Well, they were measuring the diameter of the circles to see how far the antibiotic cleared and how effective as a result it was. So the diameter of the clear zone was surrounding the antibiotic, and that's got to be something that you can measure. You can't just say effectiveness of antibiotic, because if you said that, that's not measurable, is it? Because it could be effective if it made you jump around on the spot or something like that. It's a bit of a subjective term. So we physically measure things with rulers, thermometers, that kind of thing. So you've got to give a practical example there. So now we've got a bit of a longer question. The most effective antibiotic was manufactured and prescribed to patients. 2 years later, scientists repeated the experiment with a fresh sample of the bacteria and found that no clear zone was produced, okay, which is quite important. If there's no clear zone, then that means the antibiotic is not effective on that particular bacteria. So now it's asking us to suggest an explanation for this observation and explain how this situation might have occurred. So we've got two parts to that question. We're giving a reason for it, and we're explaining how it may have happened. And we're looking at a 4-mark question here. Now, if you look at my little sketch at the bottom here, okay, basically what they've done is they've 2 years later come back to the situation and found that there's no area around the disc that's clear at all, which is important because it suggests that the antibiotic is not effective at all, okay? And the only reason that we know that that could happen is when you've got the bacteria becoming resistant to the antibiotic. So you get 1 mark for simply stating that. The bacteria have become resistant to the antibiotic. Okay, now we've got to think of an explanation point for how this could have occurred. Now, if you remember, there were some steps for the natural selection that we looked at here. There could have been a mutation in the DNA of that bacteria. Okay, that meant that the antibiotic is no longer effective. Remember, a mutation changes the DNA, which means that our specific drug can no longer get rid of or kill that. Okay, so that's for point 3. And point number 4, the resistant bacteria reproduce, this is the important thing, and they pass on that trait to their offspring. So what you end up with is a lot more of the bacteria containing the mutated gene, which means that they are all now resistant to the antibiotic, which is why you get absolutely no cleared area. Okay, so that was 4 marks. Okay, question number 3. Exercise helps to keep your body fat low and strengthens your heart. Taking regular exercise can increase your resting metabolic rate. The table lists some very different activities and how much energy in kilojoules per minute they use up. So let's have a look at this. What is meant by the term metabolic rate? So this is a classic example of where sometimes the data does not instantly help you to answer a question. So the things that you've got to say is the rate at which chemical reactions occur in the body. Now, if your specific example is breaking down glucose, as you know that occurs in respiration, then that's fine. But the key thing that a lot of you are missing out is rate. It's not breaking stuff down. It's how fast those chemical reactions occur inside. If we're being even more specific, not just inside the body, but inside cells. Okay, because as you know, metabolism is what's happening inside your cells. So Jim watches TV for half an hour and then slowly walks to his friend's house, which takes him 10 minutes. So how much energy will he use up? Lots of people got this right. Okay, so you're just taking the metabolic rates from up here. So watching TV for half an hour, okay, and then slow walking for 10 minutes. and you added those two things together. I think everybody got that right. Just be careful you put the units in the correct units. Okay. Now, Jim eats a packet of crisps that contains 760 kilojoules of energy. How many minutes will he have to jog at 5 miles per hour to burn off the same amount of energy? So he needs to get rid of 760. Okay, the reason I've divided it by 40 is because I looked into the table, okay, and I saw that running burnt off 40 kilojoules a minute. So how many times does 40 go into 760? It's 19. Lots of you forgetting the really, really important bits. Okay, silly example, but it could be 19 sausages, 19 potatoes. Like, you've got to really, really make it clear. Okay, now both of these questions were 2 marks, which means that you get 1 mark for the correct answer and you get 1 mark for the correct working. So you can't get penalised if you've lifted out the wrong data as long as you've used the correct method. So do bear that in mind, don't just do it in your head, okay? Right, okay, so question number 4. Vaccines like the MMR vaccine give people immunity to future infections. Which 3 diseases does the MMR vaccine protect against? So we've got measles, mumps and rubella. That was a fairly straightforward looking up one there for 1 mark. Now, in this question you will be assessed on the quality of your English, the organisation of your ideas, and the use of appropriate specialist vocabulary. Okay, this is one of those questions where in order to get the full 6 marks, which obviously you'd look at before you had a go at it, you have to do all of those things that they've asked you to do in the question. Now, we've looked at vaccination before a number of times, okay, and you should very much take this as a process that starts in a particular place and ends in another place. And that is how you organise your response. You start off with the injection of whatever it is that you're talking about, and you end up with how it's helping you to protect yourself against disease. So you're literally taking it in a linear fashion. So we're asked to explain it. So the body is injected with a dead or inactive form of that pathogen. Now notice I can use the term pathogen or microorganism. A pathogen is a specific type of microbe that causes disease. Okay, a microorganism, as you know, can exist both inside and outside the body and doesn't necessarily have to be disease causing. So that's an example of vocab there. For those of you that didn't get the vocab mark would have just said germs or bacteria, okay, because vaccinations, as you know, are specific normally to viruses, but actually can be bacterial too. So point number 2 comes to saying these pathogens or microorganisms are harmless but carry, another specialist term here, antigens. Now I don't know if any of you can remember what an antigen is, but if I draw you a little picture over here, okay, that's my microbe or my pathogen, and they're coated in these things called antigens, as every single cell in the body is, which is interesting. Those antigens, if you remember, are like little sort of flags, okay, and those flags signal to the immune system what that particular thing is. So if a cell arrives into the body with those antigens and they are unrecognisable, or what we call non-self antigens, then the immune system knows that they need to kick in and start fighting off whatever that cell is because it's non-self, okay? So that's another term there that you're going to have to go away and learn, antigens. So point number 3. These antigens, or flags or signals, but antigens is the correct term, encourage the white blood cells to produce antibodies. So the antibodies, if you remember, are those things that are normally drawn in diagrams as they look like Y's. Sometimes they're upside down because they can be any way up. But they're, if you like, the tools that the white blood cells use to fight that particular pathogen. Okay, so they have to make them in order to fight that particular antigen. So, next up, for point number 4, the antibodies then attack the pathogen or the microorganism, and normally the effect of that is that they will kill that pathogen. Now, lastly, the 2 points at the end that kind of relate to the question, which is how does that help protect us against disease? Well, if the live pathogen appears in the blood again, so we now go out and we come across chickenpox as a virus again, then the white blood cells produce the antibodies, again, much like it did before, but the whole point in this vaccination process is, just like making a cake, because they've made the antibodies before, they can make them much more quickly so that the illness doesn't develop. Basically, the antibodies are made much more effectively, much more quickly, and therefore all of those pathogens and microbes are killed before the symptoms start to incur. Okay, so 6 marks. You'll notice that I tend to take it as a sentence thing. And that's really all you need to do, okay? Try and keep it very, very simple and short, and don't be afraid of going outside the lines, okay? You're not allowed to write right at the bottom. We'll talk about that a bit later, but don't be afraid to use up extra space if you need to. Okay, question number 5. Now we're looking at the knee-jerk response, which you will know as a reflex arc or an automatic response. So scientists tested the reflex actions of 8 volunteers. Each person was tapped just below the knee with a small rubber hammer. When the leg was tapped, it automatically kicked outwards at the knee. The scientists recorded how long it took each volunteer to respond to the stimulus of the tap on the leg. Each person did the test 20 times, and an average reaction time was calculated to 2 decimal places, and these are the results. How can you tell that the volunteer's response was a reflex? Reflex, as you know, we use that in conversation. It's not just a science term, but a reflex is something that happens automatically. So the 2 reasons for our 2 marks, we can tell that the reaction time was really quick, can't we? Okay, if it's a considered response, we don't respond in 0.05 of a second normally. The second thing was, you're told it in the top, the knee jerk was an automatic response, which means it didn't involve the brain. So sometimes you have to look not just in the data that you're given, but also in the blurb above, okay, which is why sometimes I say have a look at the question, then perhaps look back at the big blurby bit at the top. So question number 5. Another reflex is the response of moving your hand away from a painful stimulus. Below is a diagram of the parts of the nervous system involved in this reflex. Okay, you can pretty much guess what they're going to ask if you get a picture like this. Okay, so before I do anything, I'm going to just look down. So we're looking at part X in the diagram, which is indicated there, and what it is, and describe, no explanation needed, just to say what you see, the path taken by a nervous impulse in this reflex from the stimulus to the response. So I always advise you to do this, girls, is before you start, make sure that you've got those key terms right, okay? Stimulus is interpreted by a receptor or picked up by a receptor. In this case, our skin on our hands has lots of receptors on it. The first set of neurons, remember this isn't just one, are our sensory neurons. Okay, then we've got our relay neurons here in our central nervous system. And then we've got our motor neurons, and I would in an exam just write all of this on here before I start to check that I've got it in the right order. And then obviously at the end, this is not terribly clear, but this is our muscle, and we call that an effector because it has an effect. Okay, so I've annotated that. Obviously there are a couple of other things that are happening here in the gaps. We've got our synapses, which are basically the gaps between the neurons. So you've got lots of stuff that you can talk about. And just like I told you with the vaccination thing, start at the beginning and work your way all the way through round to the response that's happening here at the muscle. So, point 1, pain stimulus is detected by the receptor, as annotated there. Point 2, impulse travels through the sensory neuron to the spinal cord. This bit's really important, okay? Remember, those nerves don't go anywhere. They're like wires in an electrical circuit. The impulse, the electrical impulse, is travelling through them to get from A to B, okay? When the impulse reaches the synapse, talking about the gap now between the neurons, it triggers the chemicals to be released. And we'll be learning about that in one of our revision lessons again if you are a bit confused. Remember, those wires aren't just joining end to end. The wires have a gap in between, which means, well, the wires being the neurons, which means that an electrical impulse cannot travel through the gap. So chemicals have to pass on the message between the two, if you remember. So these chemicals arrive at the next neuron and signal a new impulse. The impulse travels along the motor neuron, arrives for point 6 at the effector, and this signals muscle contraction. So you can't just say this signals a response, because our muscles can't obviously yell and scream, can they? You've got to say what that response is, and the point is we want the muscles to contract so that we can pull our hand away, our arm away from whatever it is that's causing us the pain in the first place. Okay, automatic response, no considered action required, no brain, much quicker because it doesn't have to travel as far. Okay, and all you girls have to do is try and make sure that you can remember which part of the pathway goes where. I've given you my advice: sensing, sensory going in. Okay, motor, it's just about to have a physical effect, so it's going out. But if you can't remember that, you need to find another way of trying to do that. Okay, question number 6. We're on cloning now, so we've done this quite recently. Got a nice little diagram there, but we don't need it to start off with. So what is a clone? An individual that's genetically identical to its parents. A lot of you just put it's genetically identical. Well, to what? To the Queen? You've got to make it clear what actually that phrase means. Clone is used to refer to 2 generations of individuals. So the new cell will be a clone of one of the original cells. Which one, A or B? So you can see in this diagram that it's got to be B, because that's where the genetic material is coming from. So I've just put B because the DNA originated in the body cell, or cell B. Explain how an animal can be cloned using adult cell cloning. So looking at the point allocation down here, you've got 4 marks. Okay, although it's an explain question, we're literally looking up here and saying again what we see. Okay, a lot of the information is actually on there. So the first thing you've got to do before you can do anything is remove the nucleus from an unfertilized egg cell. So this cell up here has had its nucleus removed. Remember, if you don't do that, you're going to end up with a mutant, because you'll end up with 46 chromosomes from here, 23 from here, And as you know, that's far too many chromosomes to have in an individual. So that's where you've got to start off with. Then, logically, you insert a complete set of chromosomes from the adult body cell into the empty egg cell, just as shown up there in the diagram. Okay, point 3, you stimulate the egg cell to divide with an electric shock, and that basically turns it into an embryo. Okay, an embryo is a cluster of cells, cluster of early cells. And then this is implanted into the new embryo, not into the new embryo, this embryo is implanted into a surrogate mother. We don't have to say it in this question, but a surrogate's used, obviously, because then you can use your perfect individual that you're cloning to make another egg. You're not having to wait 9 months or however long it is for that particular organism. Okay, right, so in this question, we're on question number 6D now. This is another organisation of ideas, use of appropriate specialist vocab, okay, and we're asked to discuss some of the reasons for and against the cloning of animals. The discussion here, because they've said for and against, okay, those two things there, that replaces our evaluate question, doesn't it? Wherever you see the idea for and against, you are looking at evaluating a concept. Okay, so I'm evaluating. Now, the organisation of ideas in an evaluation, plenty of times we've discussed this before. You should really separate the two sections out, just like I've done here, okay? English could probably be better than in my answer, so I've just written for and against, but you would probably put in front of this reasons for animal cloning, reasons against animal cloning, okay? So, and notice down the side here I've got 3 and I've got 3, okay? The reason being is because with these organised answers, you're looking for a balanced argument. So you can only really get the full marks if you do half and half, depending on how much the question is worth. So the reasons I've got, farmers can produce lots of ideal offspring very quickly. Studying clones could give a better understanding of the development of the embryo, and also ageing and age-related disorders. And most of you got this last one. It could be used to help preserve endangered species. Lots of you said extinct. If they're extinct, they're extinct. There's not really a lot you can do about it. And you wouldn't really preserve an extinct species. You'd have to resurrect them, which was obviously, it was done with the woolly, tried to be done with the woolly mammoth recently, but it's not to do with preserving species at all. It's to do with resurrection, as I said. So against, cloning leads to a reduced gene pool because they're all the same. There's less variation in that population. And if that population are closely related and a new disease appears, they could all be wiped out. It's because they're all the same. Okay, cloned animals may not be as healthy as normal ones. We've talked about Dolly. She didn't live very long. She could well have died of the respiratory disease that she had if she hadn't have been a clone, but they think that it was a result of her being a clone. So, and then lastly, it could lead to attempts at human cloning in the future. Ken, I've just put at the bottom, make sure that it's balanced. You can't have one on one side and 45 reasons against on the other side of the argument. And that's quite important. Unless they ask you for a conclusion, you no longer have to put one. Okay, so that's quite important. Okay, question number 7. You've got given a food chain here. The diagram shows the amount of energy combined with an area of plants. It shows how much energy from the plants is transferred to each trophic level of a food chain. So trophic level here, 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 here. Okay, and it's just asking you which of the pyramids of biomass represents the food chain. well, if you look up here, it's going up. The amount of energy is going up from the top carnivore or down from the producer. So must be D, that one. And say specifically because the biomass at each level of organisms decreases, as you can tell, which is what it tells you in the food chain up there. Calculate the percentage of energy in the grasshoppers that's transferred to the mice. So grasshoppers had 1,100 joules. Only 130 joules got through. So remember, a little cheat for you there. 130 divided by 1,100. The cheat being, if you're confused, it's the smaller number on top of the bigger number. Times it by 100 to get your percentage, and you got 11.8. Give 2 ways in which energy is lost within a level of a food chain. We spoke about this a lot, really. You can't just say by respiration, movement and growth. You've got to talk about the fact that at each level it's used for that. So, say the grasshopper will respire or move or grow, which means that the energy that's used for that process cannot be passed on. Alternatively, you can talk about energy being lost, but it's only really lost as heat or excreted, or as some of you put, not all of the organism will be eaten. A good example of this is foxes don't eat all of the rabbit, they won't eat the bones because they're indigestible, so a lot of the energy is locked up there. So the food chain has 4 trophic levels. Most food chains have no more than 5 trophic levels. Explain why the length is limited in this way. This was really one to sort of food for thought if you think about it, and a couple of you got there in the end, really. If you think about this can't carry on forever, can it? Because the amount of energy is decreasing or depleting each time, which means that the next one is less likely to sustain the next level of the food chain. Which actually makes quite a bit of sense, really, if you think about it. You can't keep taking the energy away, it won't appear from nowhere. So the food chain has 4 trophic levels. So energy is lost at each level of a food chain. So that's our first reason. After about 5 levels, the amount of energy being passed on is not efficient to support another level. So you say you have your top predator in the Kruger National Park. You've got your lion, okay? Lion's top level predator, consumer, can he support another level? Probably not, because by the time the energy's got to him from the producer, it's decreased so much, okay? Question number 8, natural selection, okay? Again, I'm going to pop down to the bottom to see my questions first. So, which variety of moth has a better chance of survival in a soot-polluted area? Well, you can tell by the pictures, can't you? You can barely see that black moth there. So the answer is the dark variety. Using the idea of natural selection, explain why this variety of moth became more common in soot-polluted areas. So you've got to talk about the fact that the dark moth is much better camouflaged. And those of you that got all the points here said it's less likely to be eaten by predators. That's its survival advantage. Okay, it's not that it's a colour and all of the other moths think that it's cool and therefore they like it and they don't kill it. It's to do its survival. It's always survival, and that one can survive because the predators cannot see it. So less likely to be eaten by any predators. So the dark moths survive, breed, and pass on the gene for darkness to their offspring. So you get more and more individuals with that trait appearing. The bar chart shows the percentages of dark and light coloured peppered moths in 2 different towns. Which town is the most polluted, A or B? Well, you've got more of the dark variety here, haven't you, than in this one, which means that this one was probably slightly more polluted because they're surviving more. Okay. There is a higher population of dark moths, therefore, in that area. Calculate the percentage difference between the dark-coloured moth population in town A and B. Look at your axis. That says percentage of moth population. So it's really simple to do this one because you're looking at the difference between that level there and that level there, which I made 77% minus 25, giving me 52%. Okay, the last question now, to do with the carbon cycle. Looks slightly complicated, but actually it was a nice, simple question. So describe what's occurring at point X, Y, and Z in the cycle for 3 marks. So I would make sure that you take X, then you take Y, then you take Z, okay? And try and explain them in relation to the carbon cycle. So, at point X, which is here, you've got carbon dioxide being removed from the atmosphere by photosynthesis. Please don't do that, that's just me. Write it in full. The reason you know it's being removed is because the arrow is going into the thing, into the tree, excuse me. So point Y, CO2 is released into the atmosphere by plants and animals respiring. So that's point Y. You've got carbon dioxide in the air, so you know that that's respiration. And for point number 3, at point Z, carbon compounds in plants are being transferred to animals as they eat plants. You can see here the rabbit is eating, well, it says it's eating the tree. I think that's too high up for it, but it's eating plant material. So that's how the carbon is getting in there. Okay? And how are microorganisms involved in the carbon cycle? Now, if you remember all that work we did on decay recently in our revision lesson, microorganisms are all along here, wherever you've got dead matter, dead material. They will break down the waste products and dead organisms, just like in our compost heap. And as they're doing that, they release the carbon dioxide into the atmosphere. It's not necessarily instant. It's not the grass that they're eating, they're releasing that CO2. It's more a case of they're releasing a form of CO2 because they're living, because they respire. So you mustn't forget that bit at the end for those 2 marks. Now, my advice to you from here on in is have a look back over the paper that you've just done. Figure out the questions that you did least well in and go away and form your learning, your revision around those questions now.